We immediately proceed forward with our next keynote lecture, which I urge everybody who is in the chat room to stay and listen to, because it will be very interesting. The speaker is Jeff Galvin from American Gene Technologies. Jeff, you're welcome. The floor is yours.
Thank you. So pleasure to be here with you today, and it sounds like I listened to a little bit of the last presentation, so I think this is going to be a little change of gears. We're going to talk about gene and cell therapy, which is my expertise, having been in this field now for 15 years. And a little bit about my history is that I come out of the computer industry. I was retired after having a success in Silicon Valley, and I met somebody at NIH who showed me viral vectors, which I'm sure most of you are familiar with, but just to make sure we're all on the same page, you know that viruses have been infecting your cells now for 1.5 billion years in animals, and they carry little instructions inside of them. The most recent one that everybody is aware of is, you know, viruses could carry RNA. You saw the vaccine had mRNA, gets into the cells. It's essentially like software. It tells the cells to build something new. Genes are almost like instructions to the cells. They're not coded in zeros and ones like your computer, but they're coded in ACTG. So you can think of all the stuff that's coming into your cells from viruses as like software. Well, recently, and by recently I mean really in the last 20 years or so, we've come to be able to reliably crack open viruses, scoop out their instructions and throw them away. And what we have left over is an empty stealth bomber, or almost just a delivery mechanism where we could deliver anything. So we can replace that old malevolent bad instruction with something good. Instead of something that hurts your health, it could be something that improves your health. That's the whole concept behind gene therapy. If you are familiar with computers, you can think of it as we are converting viruses into updates for the organic computer, the human cell. And now that we can edit DNA, the question might be, what can't we do? And the reason I fell in love with this immediately upon hearing it is because I was there at the beginning of the computer revolution. And I realized that a technological revolution was coming to pharmaceuticals, where the power of these things was almost infinite. And because we're talking about software, it was going to be completely different than all of the traditional drug development modalities, which are basically discover a molecule, test. If it does what you want, great, so long as it doesn't have a side effect that rules it out as a beneficial drug to the public. And this is because, you know, how does a drug know where to go in your body? It doesn't. You take an aspirin, it goes everywhere. Fortunately, aspirin will reduce your fever. It will reduce inflammation. It will, reduce your headache or other types of pain. And the side effects are minor. Even though it goes everywhere and treats every cell in your body equally, the side effects are things like thinning of the blood. Well, if you're not a hemophiliac, you are okay. So that is a normal drug that was discovered in tree bark. But, you know, modern times have only advanced it to the point where we can generate tens of thousands of new molecules, monoclonal antibodies, new, things, and then screen them down to a couple of things that are worth testing in the clinic. And 1 in 5,000 of those becomes a drug. Well, that's because of the idea that it's untargeted. Essentially, even monoclonal antibodies are only slightly targeted. And the fact that you do not know whether it has a side effect until you test it in thousands of people. Well, that's not the case with gene and cell therapy. This is software. This is deterministic. We know exactly what the gene cargo would do in your cells. We can even just test that in a cell culture hood. We can make sure we can add things to the virus that target it to certain cells, and then we can add things to the genetic information inside the software that are like an if-then statement that says, if this is true, then turn on the drug. Well, this is further targeting that isolates off-target side effects in healthy tissue. That if-then statement can detect enzymes or proteins that determine what type of cell we're going into right now. So it could determine things like, is this a liver cell? We only want to direct this to the liver. We don't want it expressing in the heart. So, we can actually put an if-then statement in there that looks for albumin. Albumin is common in the liver and not so common elsewhere. We could also do things that target it to a specific disease. So if you have high levels of prostate-specific antigen, PSA, human prostate-specific antigen, that cell probably has prostate cancer. So we could go and put an if-then statement in there that says, turn on if this cell probably has prostate cancer. Well, you can imagine now that we have, drugs that are that highly targeted, we can go ahead and put very specific things into very specific cells for very specific purposes. It's a whole new world. And my personal belief is that everything will be cured. This looks very much like the computer revolution that I joined in the 70s. And the technology just improves every year. It doubles in efficacy. It doubles in functionality. And yet the cost halves. And in that kind of environment, what is possible? Anything. If you can dream it, you can do it. Now, you won't get there overnight. In the 70s, we knew eventually computers would drive your cars, and we're not quite there yet, but we're darn close. But if you had said in the 70s, oh, if you can dream it, you can do it, they'd say you're crazy. Okay, so I promise you that this is exactly the same. I've been through many tech revolutions. I've seen many naysayers. I've seen many things happen that were highly predictable, that people, because they were so wed to the traditional ways of doing things and so familiar with that, they had a hard time embracing. But I promise you, this is not going to be an issue with gene and cell therapy over the long term. You're going to see a drip, drip, drip of miracle cures. You're already seeing that, a cure for blindness, a cure for spinal muscular atrophy. This is blind people seeing again, cripples walking. It's only a matter of time till we heal the lepers. Okay, this is the stuff of miracles. But it's not miracles, it's just science. It's just our understanding of your DNA and how it impacts your biology, our understanding of your biology itself. How does your immune system work? How do diseases move around your body? And then it's this increase in editing capabilities. Right now we have reliable viral vectors that can upregulate or downregulate any cell. We can do this in a targeted manner. There's a limited scope of diseases that are addressable today, but they're very important. And over the long term, we'll cut in, you will find that things like CRISPR become so reliable that we can use those in the clinic. Right now, the only complaint I have about CRISPR, and the only reason we don't use it, is because if you have a batch of cells and you need to change 100% of them, you can't do it with CRISPR. Because if you put enough CRISPR in there to change more than about 30% of the cells reliably, CRISPR starts making off-target cuts. So you can understand how cutting DNA in the wrong place could be very damaging to your body. You could create cancers, you could create all sorts of problems, the least of which being killing the cells. So we use viral vectors because they're more reliable. We are actually able to get in our first drug, which I'll tease you with this. It's a cure for HIV. Probably familiar with HIV. It's been 40 years on the planet. It's treatable, but there are 38 million people who have HIV. And I think worldwide, approximately half of the people take antiretrovirals to suppress the virus. In the United States, the conformance to the regimen is even lower. So only a third of the people take antiretrovirals. The rest of them are contagious and progressing to AIDS. But they don't take antiretrovirals because they're so painful. Taking daily antiretrovirals means nausea, diarrhea, and fatigue every day. You don't feel normal. The other thing that it means is that over the long-term exposure, you get early aging, bone density issues, osteoporosis, brittle bones, liver, kidney, heart disease, and extra cancers. These patients are very expensive and they're in and out of the doctors all the time, and they have a low quality of life. They are suffering. And so we're very excited to be able to bring something to them that we think will be a one and done treatment where we will modify their immune system to make them completely immune to HIV. In other words, their natural immune response to HIV will be such that they can never get AIDS, they can never infect another person, and they can never contract HIV. And we think this will be durable for life. One treatment, lifetime normalcy for somebody that right now is suffering a very abnormal life. But this is the power of gene and cell therapy, is that HIV came into scope, but eventually everything will be in scope. You can imagine that if you can cure HIV by modifying the immune system, why not cure hepatitis B, HTLV, Epstein-Barr, CMV, human papillomavirus, herpes? It's all just a matter of cost. Well, remember, functionality up, costs down, all of a sudden everything is practical. Why suffer cold sores anymore? If we have a cheap treatment that we can go ahead and make sure that you'll never get shingles, you'll never get herpes outbreaks ever again, why not take it? It's just a matter of cost. So this will flow through the entire world. It will not just eventually cure everything, all cancers, all chronic viral infections, all viruses, all monogenic diseases. If you're born with a monogenic disease, what are you born with? One broken gene. You're missing a gene somewhere in your body that you need for a normal life. Now, this could be irritating, lactose intolerance, or deadly spinal muscular atrophy, where people that are born with spinal muscular atrophy in the past, by the way, were guaranteed to die in a iron lung, with a breathing machine by age 4. Well, there are 8-year-olds now running around that were born with spinal muscular atrophy. Because what could be simpler than ordering the gene that you're missing off the internet? Comes as a plasmid. You go ahead and you crack open these viruses, scoop out the insides, get rid of the bad stuff, and you put that gene in there and you infect a cell, and now it has that gene. It's not a cure. You're just not sick anymore, right? I mean, the point is you're returned to normal. That is, the best cure, returning you to a natural state where you're no longer threatened by whatever it was that you were born without. With HIV, we learned a long time ago that people that were born with a broken CCR5, chemokine receptor 5 gene, had high resistance to HIV. We've learned a lot more since then. There's been bone marrow transplants where people who got bone marrow from people that had that gene defect, essentially, missing CCR5. We could do a bone marrow transplant to somebody that had HIV, and they could naturally control their HIV after that. Now, that was limited to certain strains of HIV, but we've learned a way to make it applicable to all strains of HIV, and that's what I'm going to talk about now. So it's a revolution in pharmaceuticals. You might think about how this impacts everything that we're doing in healthcare. There's going to be a lot of opportunity. There's going to be a lot of disruption. It's all going to be a positive thing. The drug market will turn from $1 trillion a year into $10 trillion a year pretty quick. That sounds like a good thing for pharmaceuticals. But of course, pharmaceutical companies are huge organizations that have a hard time essentially adapting to new technologies. So it may also be a painful transition for some companies, but it will be for the good of everyone, a bigger market for pharmaceutical companies. So that's good news for them. But for patients, it means cures where there were only treatments before. So let me give you some examples at AGT to kind of put this thing in context. I'm going to go ahead and share my screen. Hopefully this will work. Yeah, it looks like you are now seeing my screen.
It does work excellently, thanks.
Okay, good. So now let me just see if I can see everything that's going on. Great, I can see now. So first a disclaimer, because I'm going to say a lot of forward-looking statements here, and it's important that if you're watching this presentation that you understand that not everything I'm saying is guaranteed. As a matter of fact, nothing's guaranteed. So just take that, as a warning. Don't run out and use this information improperly without doing your own research and understanding it more deeply. So AGT right now has 3 lead programs, one in HIV, one in phenylketonuria, and one in solid tumors. Now, solid tumors, what I mean by these are epithelial malignancy, which are really important: breast, prostate, lung, liver, colon, kidney, ovarian, pancreatic, head and neck, and skin cancers. 900 people a day die in the US from those cancers. In the US alone, 900 people per day. Okay, and it's expensive for them to die. So it's not like we get out of the cost as a society, because in the United States, of course, most people are insured. So the insurance company will spend about $500,000 on them dying. So this is quite a burden. While you're treating these people and trying to extend their lives, you're spending a lot of money. So these are all really big markets, really important markets. Can you see my mouse here? You see it when I'm moving my mouse along?
It is visible, yes.
Okay, good. That way I can point to the slide. So you can see it's a very big market in any case. So here's how we're curing HIV. And after you see this, you'll go, aha, why not? Remember I told you that if you transplanted bone marrow from an immune person who's naturally immune to HIV into a person with HIV, it will suppress their virus durably. As a matter of fact, there's been an example of that. The Berlin patient unfortunately passed away really just from age-related diseases, not AIDS-related. He literally lived without antiretroviral therapy for almost 2 decades. And we have other people that have lived almost that long from other experiments, one that I'm going to talk about later. So we know that if we can modify your immune system so that it doesn't have that latch point for HIV, the HIV T cells can do their job, which is come to the viron and kill it. The problem right now is the HIV viron will actually grab onto the HIV T cell, merge with it, implant its DNA, and the sentinel T cell that was there to detect and begin the immune cascade against HIV is now the carrier of HIV. So that is the problem with HIV. But if it can't get in, the HIV T cells work normally and clear HIV like you clear a cold. The concept is so simple, and it's proven out by the Berlin patient, the London patient, the Amsterdam patient, and even gene therapy experiments where they removed enough CCR5 to get durable remission in a number of patients. So what we've done is we've taken a giant step forward on all that research, and we've been able to make a practical treatment at a cost where it's cheaper to cure an HIV patient than to treat them for life. And the way that we do it is we draw a leukapheresis, which is a filtration of the blood, about 300 milliliters of blood. So what happens is it essentially pulls blood out of one arm, filters out T cells, puts the blood back in the other arm, and you come away with a lower amount of T cells after the leukapheresis, but it's not so much that it causes you any problems, even if you have a low T cell count. So we do a fairly simple blood draw. And the reason that we use leukapheresis is the part we're interested in is the T cells, of course. So the next thing that we do is we put it in an automated benchtop unit. This is important because this needs to be scalable, it needs to be automated, and it needs to be distributable around the world. A $250,000 machine, which is affordable by any hospital or clinic that treats AIDS probably on the planet, will do all of the processing automatically, just getting reagents from American Gene Technologies. So the first thing that it does is it stimulates all of the T cells that are in the blood draw. And we stimulate them with something that activates the HIV-specific T cells, the CD4 positive T cells. These are the conductors, the detectors and the conductors of the immune response, and it proliferates them a little bit in the mix. The next thing that we do is we deplete them, all of the T cells that we don't like, because it doesn't matter if your cold T cells are immune. What matters is that your protection is immune. So we're only interested in your T cells that are HIV specific, so we get rid of the rest of the T cells, which makes the next part much cheaper because we use virus, which is very expensive, in the mix to modify every single T cell in there. And the modification that we're bringing is we're stripping them of CCR5. So we're giving them this magic mutation that some people in Northern Europe are born with that makes them immune to most HIV. But we're also putting in additional protections that work on non-R5 strains. So all known strains of HIV now, we have 3 barriers of protection. And what we're seeing is 95% protection of all of these T cells, which means that most of them are able to kill HIV instead of being killed by HIV or utilized in HIV to spread the virus around. The last thing that we do is we culture them up to 1 billion cells. Why 1 billion? Well, 1 billion is 10 times the number of CD4 positive T cells that you have after clearing a virus. So once you clear a cold, you have about 100 million cold specific CD4 positive T cells. So we thought 10x on that would be pretty good memory level protection against HIV that should be able to overwhelm the HIV and overwhelm all the infected cells in your body that are producing HIV virons and suppress the virus for life and eventually eliminate it totally from your body. But in the meantime, because it's a functional cure, which means as good as, you could think of it that way, or equivalent to, it's suppressing the virus at levels where you're not contagious. That's one critical factor, right? You want to return to normal life. You want to know that you're not a threat to people around you and your loved ones. And also you want to get off of that antiretroviral therapy that makes you feel sick. And also it's just a reminder every day that you're different. The next thing you want to know is that you can never get AIDS. Well, if the virus is suppressed at that level, you can never get AIDS. And then the question would be, how long does this last? And it looks like these T cells will last in your body for your entire life. So 1 billion of them is a great starting point. We think we can reduce the number later. After we've got 1 billion of your T cells, because we're actually taking blood from an HIV infected individual, getting these T cells and making a personal blood product, an autologous cell therapy to put it back in them. There's a extensive set of tests that make sure that the modification worked properly and that the cells are healthy and that there's no contaminations. And the final step back into the patient's body takes about a couple of weeks for these cells to do what's called engraft, get used to your body, recover, get ready to be active again. Then they go off the antiretroviral therapy. And what we expect to see is what's been seen in previous experiments when they've been successful, that it suppresses the virus at levels where you're no longer contagious and you don't need antiretroviral therapy or any other therapy to keep it at that level. So that's what we're going for. And you say, well, if it's been done before, why do it again? Well, the difference in this therapy is that it's 1/10 the cost and much more reliable. Okay, we're actually converting 95% of the cells to immune instead of 10% of the cells. So you can see that the overall commercial potential of this is much higher because we can bring the cost down to something where this would make sense for anybody. But I'll talk about some other things that we've improved on in just a moment. But here's why I'm so confident that this is going to work. 1 billion T cells is almost 3% of your circulating T cells in your body at that moment, of your CD4 positive T cells. So the chances of an HIV viron meeting a T cell that can kill it is very, very high. The chances of HIV meeting a T cell that cannot kill it is very, very low. And that is because it's your HIV specific T cells that are searching around for the HIV viron. The ratio will be literally 1,000 or 2,000 to 1 of immune cells that are immune to HIV, that are clearing HIV, to ones that aren't. And remember, the CD4 cell, if it stays in your body and stays active, it isn't the only fighter. It actually creates an immune cascade that creates CD8 cells, which are uninfectable by HIV. The only reason you lose your CD8 cells is because HIV eliminates all your CD4 cells. But with a normal CD8 response, you get further protection, and then you get an antibody response on top of that. All of that is due to the way that the CD4 cell detects and alerts the rest of the immune system. So that is the pivotal thing that you need to restore. And what we've shown is that we can restore it at levels where this blood can clear HIV better than anything that's ever been seen before, including antiretroviral therapy. So we made blood in the preclinicals from HIV infected individuals. We went ahead and we tested that blood in a cell culture hood, in a cell experiment. So in a beaker, we can dump as much HIV in there and as many infected cells as we want, and it sterilizes itself of HIV. And when you do a calculation, what you see is it looks 3 to 5 times more potent than it needs to be to clear the entire body. And the results were so remarkable that we showed them to NIAID, the National Institutes of Allergy and Infectious Disease. Many of you have heard of Tony Fauci, and now that he's famous for COVID, but he's more famous for HIV, believe it or not. That was his lifelong career. And his lab is run by a guy named Tae-Wook Chen, and he was so impressed with our experiment that he wanted to repeat it in his own lab to validate it. And he was able to do that. So we just gave them the reagents, told them what the protocol was. They bled their own HIV patients. They made the same blood product. They did the same HIV challenge test, and then they wrote an article showing that it works. You can read this article in Molecular Therapy, and if you have a background in HIV, you'll be convinced, like everybody is, that this is a giant step up from previous experiments in terms of reliability and potency, and that it should be enough to give a functional cure to perhaps all the patients to take this. It may actually hit 100%. So remember I told you that somebody tried this experiment before where they stripped CCR5. That was a company called Sangamo. So Sangamo was able to get 1 in 10 patients in durable remission. 1 in 10 patients. That's not bad. The problem was that the way that they do it is what's called zinc-fingered nuclease clipping of genes, which meant that you had to get 2 molecules into each cell and hit 2 different sets of chromosomes to get rid of all the CCR5. So in the best of cases, they could only get 10% of the cells immune. But they got lucky with 1 out of 10 patients in the first experiment, 1 out of 12, and that person went into durable remission. They have been without antiretroviral therapy, living a normal life for 8 years now. So that's remarkable, right? This is what we call a functional cure. You could, if with a very, very sensitive experiment, you could find some HIV in this person's body, but they can't infect anybody. So it's like, you know, herpes that's in remission, right? You can kiss your wife or your husband and you don't pass herpes unless you're having an outbreak. Well, they are naturally suppressing the virus better than antiretrovirals do. And so they're completely non-contagious. They can't get AIDS. And as far as we can tell, this is durable for life. But 1 in 10 patients is not enough. And also what they found about that one patient in the first experiment, they're what's called an heterozygous delta 32 mutant. That means they were born with 1 allele, 1 of their chromosomes was missing the CCR5 gene already. So it was very lucky that 1 in 10 cells was actually modified properly, and that in some cases that was enough. And this guy, they redid the experiment with all heterozygous delta 32 mutants. They only got 1 in 10 still. So they cured another couple of people, but at $500,000 worth of vector, and you got to run this on 10 people to get 1 cure, that's $5 million worth of vector. That doesn't even include all the cost of processing and all the cost of delivering this. It's completely, uncommercializable. This is not a practical solution that anybody could afford. So what we're in the situation of with their experiment is that it's a great proof of concept or proof of principle, but it's not going to be practical. Well, we've gotten the amount of vector in our process down to $35,000 worth of vector per patient, and we're delivering 2,000 times the number of immune cells as the best case in Sangamo. So for every 1 cell in a Sangamo patient that they were able to create that was CD4 positive, HIV specific, and immune to HIV, we create 2,000 at less than 1/10 the cost. This brings it into the commercializable range. But then the other thing is we're using viral vector knockdown of CCR5. We only need to get 1 copy of the molecule in each cell. It knocks out both alleles simultaneously. So this is 95% of the cells are completely immune instead of 10% of the cells, with the same number of molecules, same number of viral particles getting into the cell. This is a practical solution at this point. Plus, we're throwing in 2 other levels of protection that they don't do, which are knockouts of conserved regions of HIV that give them further protection. So that's why I'm so confident. How am I doing for time here? Can I ask the host, can I present for 5 more minutes and then take questions?
I think 5 more minutes is acceptable.
Okay. I just want to give you a sense of, another application of this. Remember, with our HIV cure, we have a platform of components that we can mix and match. We just isolate a different T cell and we can go after hepatitis B, HTLV, herpes, Epstein-Barr, all these different chronic viral infections should fall to a similar strategy, slightly different modifications to the T cells to make them immune and more effective against their disease. But the concept is good, and we have patents on all of these different steps of the process that make it practical, that reduce the costs, that expand these things, in a low-cost way, and that make it distributable and scalable throughout the world. So we will find practical solutions to all chronic viral infections. But monogenic diseases, we have another lead program in phenylketonuria. We have very good proof of principle that will work. We have mice that are cured of the that have the same phenotype as humans that have been cured. And we can advance that into the clinic eventually when we have more resources to simultaneously do many clinical trials. But another thing that we're advancing towards the clinic is a cure for epithelial solid tumors. What do those look like? Well, your body is full of epithelial malignancy. Everybody on this call has cancer, but there's nothing to worry about because you have immune cells that surveil for small malignancies. They're called gamma delta T cells. And what they will do is eliminate one or two cells that are malignant so they don't grow into a solid tumor. But if you age and your immune system goes down, or you have more, accrued stress on your body, you may get a critical mass of these cancer cells that are growing faster than these T cells can eliminate them. That is what we call cancer. 2 out of 3 people will not get that cancer because your surveillance system is just that good. You will live long enough that you will die from heart disease or a car accident or something like that instead of getting a solid tumor of epithelial type. Remarkable, but 1 out of 3 people will need to be treated for a potentially deadly solid tumor. Okay, all it is a collection of these cells that are growing faster than your body can eliminate them. But what we do is we have a viral vector that we can put into that tumor that converts a bunch of the cells to secrete a stimulatory small molecule that clings to the microenvironment of the tumor itself, and it attracts every gamma delta T cell in your body. It not only attracts those T cells, and we have to light up this tumor by producing enough of this stimulatory small molecule. It takes about 20% of the tumor cells to do that. That's reachable with an intratumoral injection. So imagine you get prostate cancer, and they say, okay, we need to palpate the tumor, or you get breast cancer, we need to palpate, find that primary tumor, and get just a normal syringe into it and squirt in some viral vector. How much viral vector? Maybe $50,000 worth of viral vector at today's manufacturing costs. All right, so not a very expensive thing. That lights up the tumor where it attracts every gamma delta T cell in your body, and it activates them and proliferates them. It's like a vaccine. It's not a vaccine, but it acts like a vaccine because it attracts the T cells that are responsible for eliminating malignancy, and it activates them in a way where they can eat at 300 to 600 times their normal rate. Well, what happens? Well, as you'd imagine, they turn on that tumor and they kill it. Now, you notice there in these cells, these dead cells over here that I'm showing you, some are red, some are green. You see, because the gamma delta T cell is amazing. It has an NK receptor that when it's in an activated, stimulated state, the NK receptor will test every cell in the area. So it doesn't care what is creating the stimulation. It's the NK receptor that's exquisitely specific to malignancy. When that triggers, that's when the cell becomes cytotoxic. So it kills every cell in that area, even if it's not secreting the stimulatory molecule, provided that it shows a set of stress molecules that indicates malignancy. So you can stimulate these things up to a tremendous level in your body. They won't attack anything but malignancy. They're completely safe at very high levels, and they are very effective once you stimulate them up like this. So they kill the solid tumor. That's good, but what about the metastases? Well, they're circulating around your body in this stimulated state, and they don't know the difference between any of these malignant cells. You now have a level of clearance, sort of a flushing of your body, where these cells will find any malignancy that is smaller than the primary tumor, because they'll stay activated while the primary tumor is getting eaten away. It becomes like a timer. And they're clearing malignancy all over your body because they're in this stimulated, activated state. They get the secondary tumors and the metastases with no further treatment. We have a 1,000-mouse study that proves this already. We can put your gamma delta T cells in a mouse. It doesn't misrecognize a single cell. Everything but human malignancy is completely invisible to gamma delta T cells. Now, they don't do much when we give you, when we have xenograft mouse model, we put your gamma delta T cells in there. They'll slow the cancer down, but they won't get rid of it. When we put our viral vector into that tumor, gone. But we can also put multiple tumors of different epithelial types, breast, prostate, liver cancer, in different parts of the mouse, treat one tumor, and all the tumors disappear. They're all about equal size. Okay, what this says is that even non-homogeneous tumors of epithelial types should be completely cleared through this method. And in fact, if we treat you for breast cancer or prostate cancer, let's say you had a little lung cancer and you didn't know about it yet, or a little bit of liver cancer and you didn't know about it yet, this treatment would mop that up. It's almost like a rewinding of your cancer clock. 1,000-mouse study, 85% of the mice come back in complete remission. This should translate beautiful to humans. This is behind our HIV project. It's not in the clinic. The HIV project is in the clinic. We've already treated 3 patients. We're already getting sufficient safety data to start open enrollment. And remember, all of the HIV patients have HIV. So we're not just going to get a safety readout from this. We're going to get an efficacy readout as well. Next year, we'll be withdrawing them from their antiretroviral therapy, and keep your fingers crossed. We think that they won't rebound. If they don't, their virus doesn't rebound, they don't need antiretrovirals anymore. They are functionally cured. We should know that by next summer. This is really exciting times around the company. Well, this program of epithelial malignancy, this gamma delta T cell stimulation program, looks to me very much like HIV did a couple of years ago. And our collaborators, the Stanford Medical School, if you've heard of them, in California, and we have one of the world's foremost experts on liver cancer who will be doing a proof of concept of this in his clinic, we believe, in 2023. So a couple of years away from now, you may no longer have to worry about breast or prostate cancer. Can you imagine if you found out you had a metastatic breast or prostate cancer and your urologist or your OBGYN said, yeah, come in tomorrow, we'll give you a shot, and there's no surgery, no radiation, no chemotherapy. Your natural immune systems is so effective in that stimulated state of clearing them that you need no further treatment. What's better at finding a malignancy? Chemotherapeutics that are poisoning everything in your body, or a homing T cell that is in an activated state that will only find malignancy and only kill malignant cells? Obviously, the side effects are much lower. The dream of this company when I began it, which was 13 years ago now, was to send radiation and chemotherapy the way of bloodletting and leeches. We may actually be able to do it. So I wanted to tell you about that. That's a future thing. That's a second act for AGT. It also is an amazing proof of the power of gene and cell therapy, that we're able to, not only, cure something like HIV, which would still be the most sophisticated gene and cell therapy cure ever produced, but that we can go on and we have a likely shot on goal with something that that is a disease of 1 in 3 people on Earth, cancer. This touches everyone. There's not a person out there that won't be touched themselves or a loved one touched by these cancers. And I think we have a really good shot at this. But gene and cell therapy is really unique. It's like software. It's like the world that I came from, which means that even if you don't get all the way there with your first try, you can debug that. It's not a throwaway like a normal molecule, right? You can't debug a new chemical entity. You don't know which, exact atom in that molecule is causing you the trouble. But when you're looking at ACTGs and you're looking at a genetic construct, you can figure out how to increase the potency, decrease the side effects, debug what you have. That's actually been done with drugs already in gene and cell therapy. So it's a whole new world. I hope I've excited you about the potential. Hopefully you have some questions for me. Hopefully we have some time to take them.
Well, it's a yes to everything. So you both excited us, and we do have some questions. But first, I would like to say that I really admire your philosophy about, let's say, experiencing your dream. And I think a lot of the great achievements in our history are achieved by people who actually already they have been there. So it's only a matter of time until those steps are, let's say, fulfilled, until the final goal is reached. And I would also like to say that today, early in the morning here in Bulgaria, we had a panel on HIV, and only reason they weren't together with your discussion was the time difference. And at the panel, we also had a few patient advocates who greatly expressed one of the key concepts in your philosophy that maybe the healthcare system is interested as a whole in other outcomes that are different from what the actual patients want.
Right.
The patients might not want, for example, 5 or 10 years extended agony, but rather maybe shorter life, but with living as a normal person, not in constant pain.
Can I make a comment on that? Because
Yeah, sure.
you're absolutely right about that. And as a company that's trying to cure HIV, we deal with all of the status quo. We've been in and we've met with all of the pharma companies. And the problem is that their models don't understand how cures can be profitable. And sometimes an economic incentive, in other words, making money, doesn't always, despite what Reagan and trickle-down economics economists would tell you, sometimes what makes the most money doesn't do the most good. Okay. And that's the problem, is that from a different perspective, the patients want one thing, but the drug companies have an economic incentive to provide something else. But here's their surprise that they're going to get. There's more money in curing HIV than there is in treating HIV. They just don't realize it yet. Okay. And so we can drag them kicking and screaming to making more money and doing more good, because gene and cell therapy, will go ahead and address a market of 40 million people. This is decades and decades of delivering these cures. And the net present value of a cure to the pharma company is more money than the lifetime of treatment. They don't realize that. My degree is from Harvard in economics, so I always look at it that way. And I have to sell based on what's good for you, right? I can't sell what based on what's good for me. That's not even a good sale. I might be able to convince you. And how do the pharma companies convince you? Well, here's your one choice: buy or die, right? And that's not a choice. That is, you're a hostage. Well, that works out great economically for those companies, but I think we can make it work out great economically for those companies by letting you out of jail, right? HIV, it was a miracle when it was converted from a death sentence to a life sentence of taking those drugs, right? For once, you now had a choice to live if you were willing to live in pain, right? But then they're in a life, it's a lifetime in jail. This is a get out of jail free card. This is what the patients want. Now, how do the drug companies make more money? You're not eliminating the cost of the treatment. No, you're increasing the cost of the treatment, but you're eliminating the side effects of the treatment. And it's not just inconvenience. Remember, these patients are in and out of the doctors all the time. They actually cost $100,000 per year for insurance companies. Only 20 to 30,000 of that goes to the drug company. They could get a much bigger fraction of that by eliminating those side effects. The insurance company will reduce their costs. The pharma company gets more money. The patients get what they want. This is the beauty of gene and cell therapy. That power would allow a new model in pharmaceuticals where, how you kept your company big? You provided more and more solutions. That's how we made Apple big, right? I was at Apple when we launched the Macintosh. We sold 90,000 of them in the first quarter, which is a very disappointing number at the time. But do we turn around and go, okay, we have 90,000 hostages now, let's just up the price and we'll make more money? Because that's drug company thinking, right? No, we said, let's pack more power into it. Let's make it more useful. Let's bring in more solutions. Let's partner up with developers so that we have, when you buy a Mac, you can not only do your word processing, your spreadsheets, but it has games and this and that and, all these productivity tools. And let's, just build the need for that core technology by making it a solution to more and more stuff. I don't think that Roche would mind being Apple right now, right? Or Microsoft. There's a lot of power in innovating and producing more, really high value solutions for your target audience. And that's how you should grow in a competitive market. And this will open that up for pharma companies and biotech companies to actually light the fire under them. Because if the pharma companies don't do this, the biotechs will. Who's going to turn down a cure for HIV or a cure for cancer? Nobody. Are they going to go ahead and take Avastin still? Avastin, gives you 6 weeks of extra overall survival if you have a a terminal cancer that is where it's on label for that. So you pay $125,000 and you get 6 weeks on average of extra overall survival. Okay, 6 weeks for $125,000 or 5 years for $300,000. Which will you take? Right? This is all.
I think we can already see this quite clearly in the example of, for example, HCV therapies, which are already capable of exterminating the virus completely. So we can see that making this choice is quite possible.
Yeah, well, the problem with hepatitis C was that it taught all of the pharma companies the wrong lesson. Because what happened was Gilead got the cure, and it was expensive to develop because it's a small molecule, right? So they invested all the same amount of money. Well, gene and cell therapy is much cheaper to develop, by the way. But here's the issue, and this is what taught them the wrong lesson. All of a sudden, Gilead's stock went from 60 billion to 160 billion because they were selling so much hepatitis C cure. But then the market ran out because they were curing their patients, and it came down again. They remember this, but they didn't remember this, right? Because how do you sustain your company? You keep on bringing in new cures that sustain that growth, and you don't worry about this part because you cure all the patients. But are we going to run out of healthcare problems on the earth anytime soon? No, there's plenty of things to cure. But you know what they say? Cures don't work financially for their companies. They actually reduce their incentive to find cures. It's crazy talk if you really analyze this thing. But unfortunately, I've talked to pharma companies that just said the cure will never sell for HIV. And I said, well, why? And they said, because antiretroviral is so cheap, you'll never be able to sell the cure. Really? Most of the cost of HIV is the $50,000 to $80,000 a year of taking antiretroviral comorbidities and side effects. You can eliminate that $20,000. You can drive it to zero. The insurance companies still want to cure the patients, and the patients still want to be cured. All the pharma company does is eliminate their profits. So that's not the way it's going. It's going that if a cure works, it's going to be adopted.
And patients nowadays are more and more engaged in the process and influencing overall policies. So I think in this kind of context, the situation is completely different.
Yeah.
So I want to now give the word to Rado, who was the moderator of the previous panel. He would like to ask you a question, and then maybe I have one final question. And we can wrap it.
Sure.
Rado. Thank you. Very interesting talk, Jeff. I have the privilege of, I was around like an hour ago, and that gives me the technical ability to stay here and to raise my question directly, as opposed to during through the moderators. Specifically, the direction in which you went in the last 3 or 4 minutes about new models and about what works and what doesn't financially. A long story short, my question to you is, and I'm sure that you have thought about it, do you believe in universal healthcare?
Yes, I do. I think for base healthcare, I think it makes a lot of sense. So, you're really getting into a part of my philosophy, which, people don't really enjoy hearing from me because it's more political than it is, sort of healthcare oriented. But what is the purpose of government, right? The purpose of government is that it is a union where all the common people are required to be members, and it's supposed to represent their common needs. And what does it provide? It provides things that the entire populace needs, and it's efficient at doing that because big organizations are never really efficient. We know that smaller, nimbler organizations are more efficient, but the reality is if everybody needs something, the difference between a big government entity and a big for-profit entity is the profit. The efficiency is the same. And if you don't believe that, we have a big company in the United States called Comcast. Comcast is most of the internet in the United States. Let me just say for the record, they suck, okay? The US Post Office is way better than Comcast, way more customer oriented. And the reason is because the head of the post office is really the president of the United States, and we elect him. But if we don't like the president of Comcast, hey, tough luck. The only cable to your house is a Comcast cable, and it's buy or die, all right? Live without internet or pay whatever we say. That is not good for the general public, right? So this is my philosophy of government. Now, do you need health care? Yes. Every human being is going to have some sort of, threats in their life, some risk in their life. It could be a car accident or it could be a disease. But the point is, basic health care, the cheapest way to provide it across society, the cheapest way to give insurance to everybody is to eliminate the profit. A one-payer system, universal health care, whatever your country can afford, right? And in most Western countries, you can afford pretty darn good health care, all right? You can always argue, wow, it could be better if there were incentives, right? If doctors made more money, doctors would be better. I don't know that that's necessarily true. Who would become doctors? People who care about people, not people that are looking for money, right? Okay, so, the bottom line is that you look at the insurance industry, 15% profit. That means we could do the same thing. Universal healthcare would cost 15% less. It's a $10 trillion global market. In the United States, it's $3.5 trillion. So I can save you $500 billion just by eliminating all insurance in the United States. You can't do it overnight, but I think philosophically, as an economist, I can tell you that if everybody needs it, the most efficient way to provide it is through government, and then to put pressure on the system through political means to make it good, right? Voters, assuming that all of these countries are actually democracies that I'm talking to right now, voters determine who runs the show, and that person is responsible for running a good show that does good things for their constituents. Otherwise, they're going to get voted out. It works in the United States mostly. We're having a real problem with democracy right now. Again, this is something that people don't like hearing me talk about, but can we get it back together again and become, a really strong democracy? I think that's likely. And in a strong democracy, yes, universal health care to me is a no-brainer.
What about at global level, not at country level?
For global problems, it's a global community. For COVID, we must inoculate everybody on Earth. We must get the vaccine out there, or we will be dealing with deadly pandemics every year forever. Okay? We're interdependent. At the United States border, we can say, we don't need to invest in roads in rural Africa. Doesn't affect us, right? So we can go, that's their problem, and our infrastructure is our problem. But when it comes to a global pandemic, we're all in this hot water together. We're either going to cool it down and we're all going to get out together, or we're not. And if people in the United States or in any Western nation think that you can fix the COVID problem without fixing it globally, you're dealing with something like polio or smallpox. It's going to keep coming back and coming back and coming back. Thank goodness this only kills, 1% of the people that it infects, or a little bit more than that. But, losing 1% of your population every year to one virus is a lot. Even in the United States, people would be screaming. They'd finally go, yeah, everybody should be vaccinated. I mean, it's really amazing to me that it's a political thing whether you get vaccinated. The science is so crystal clear on this. The risk of vaccination is almost zero. The risk of being unvaccinated is high. Which one are you going to choose? Anybody who is a scientist, anybody who's smart, your chances to go to get a vaccine in the United States, your chances of dying from the car ride to the vaccine site is higher than dying from the vaccine. Your chances of being injured in the car on the way to the vaccine site is higher than being injured by the vaccine. It's just craziness that anybody questions the value of a vaccine, and yet we have like 20 million people in the United States that are anti-vax. Okay, a rational world, I'm an economist. I live in a rational world. The real world is not rational. I always like to say, and quote me on this, maybe it'll become famous one day. Reality on Earth takes place at the intersection of nature, which is completely unchangeable, and human nature, which has the ability to deny, deflect, delay technologies, science, reality, all of that stuff, right? We live in our heads in the United States. We think reality television is anything more than television. There's no reality to it, right? Yet we had a reality television president because there were enough people that were willing to accept the idea that you can dominate the virus by thinking the right way and being a winner, right? Oh, come on, nature doesn't work that way. Eventually it'll break through.
This is so true for developed countries, I think. And sometimes, when I have the plant, for example, cut of water supply to my home for, let's say, 2-3 hours one evening, then I remember somehow, not remember, but then I can imagine what it is to not have water for some time. Then I remember. Then I realize how insubstantial some of the modern day problems and concerns and fears are.
That is a very important philosophy, is that it's critically important that, one, we have empathy for others. The best way to raise humanity is to be inclusive of humanity. Humanity stretches beyond your borders. Okay, we can't do everything all at once. And even in the United States, rich people get things before poor people do. And yet in the United States, poor people have it better than they do in sub-Saharan Africa, right? But it's to remember the people in sub-Saharan Africa who don't have water, who don't have electricity, who sometimes have HIV and can't even get antiretrovirals so that they can live long enough with headaches and diarrhea, right? And the amazing thing is if they can get their drug in Africa, this is a really hilarious thing. And I think it has to do with another concept, which is very important: gratitude for the good things in your life instead of always lamenting about the bad things. Remembering that the glass is 95% full, not 5% empty. But yet I meet people all the time that are just like, it's 5% empty, right? This is a whole concept that I try to get across in the corporate culture at AGT, is that you can feel it when you help people. That, that instead of feeling sorry for what you don't have, when you experience what other people don't have and you lift them up, the gift is really to yourself, right? If you have empathy and you have gratitude, right? It's amazing.
Yes,
money isn't even as important.
And this philosophy is sometimes gets to a point that empathy becomes a little bit egoistic in a way.
Oh, sure. Oh, absolutely. Yeah. No, I could talk for hours on this subject. Like I said, look, I love human beings, and I also understand, because human beings are quite glorious and quite dangerous at the same time because of psychology. So I think of myself as an amateur psychologist. But here's an interesting observation. People in Africa that can get access to antiretroviral therapy almost always stay on the antiretroviral therapy. Whereas people in the United States who get on antiretroviral therapy, half of them stop it. Now, why is that? Is the effect different in Africa than it is in the United States? No. It's that if an African gets diarrhea and feels tired, they have diarrhea and feel tired all the time already. They don't care about that, right? They're not experiencing a 5% emptiness in their life. What their experience is the gratitude of, I'm going to be here for another 10 years, right? With my family, right? This is a rearrangement of thinking that we can make in the Western world that will reduce our materialism and allow us to live a really joyful life on less destruction to the planet, more sustainability. I mean, it all fits together, right? But converting people's psychology can be very hard, especially against messaging that tells you, be selfish. Yeah, your problem is immigrants, right? I mean, and people believe that stuff, right? In the United States, we're all immigrants.
Yeah.
Right? So, this is just, it's craziness, but it's manipulation of psychology. Politics will tend to do that. Capitalism will tend to do that, right? Because it's really self-centered. Politician wants to be reelected. Money helps them get reelected. A capitalist wants to get rich. one way to get rich is to take some of that money and put it over in politics and cancel out all of the, the good that government could do for the general public to moderate capitalism. The best investment that a capitalist can make turns out to be investing in lawyers on K Street. These are what are called lobbyists. Studies have shown from good universities like Princeton that the return on $1 on K Street is 700 to 1. Where else can you get $700 on your $1 investment? Invest $1 million, get 700 million back. In what? Legislation that benefits you at the expense of the public. Right? It's human nature. Human nature ultimately will fall to nature. If we destroy the planet, no problem. The planet will go on without us.
It will reset itself very finely.
It will reset itself, exactly. It's been there before. A volcano will go up, cause a sustained winter that will kill off most of the humans on Earth, and the planet will recover over the next 100 million years, and life will reemerge. It may not look exactly like us, but there'll be another dominant species that will have an opportunity to maybe live longer than we just did, if they're smarter. Hopefully, they'll have bigger brains.
Jeff, it has been honestly a pleasure that you are the person to close today's session, and it's been an honor sharing your point of view, because it's not only hearing a lot of the concepts and the philosophies that drive you, but it's also sharing quite a lot of them. So thank you very much for joining the event. I really hope that through our event and similar to it, and also through the evidence that I see you are constantly piling up to really empower your concepts, you can reach and inspire more and more people to make the world a better place. This is also the main philosophy of the Biotech Atelier. We are performing every year to bring people that think likewise, that live every day in the evidence-based approach, and are eager to share viewpoints and sometimes to enter into conflicts and let's say
debate
at the bottom of each and every problematic. Biotech Atelier is the environment just for that. I will be really glad if you join us again next year, or maybe at some of the satellite events, because throughout the year we are organizing focused events. And also yesterday we had an interesting talk that touched on gene therapy as well. So maybe gene therapy will be a topic for one of our satellite events, and there we can select and bring together more people with, let's say, focused knowledge and perspective in this specific direction that I hope we are getting sooner than we can pessimistically expect.
Well, terrific. It's been a pleasure talking to you folks, and thank you for inviting me, and I hope I'll see you again soon.
Sure, we keep in touch, even offline the events. I will keep to monitor your progress and exchange further ideas with you.
Oh, well, please. I always forget to say this, but we're American Gene, American with the N, and then Gene, G-E-N-E. Okay, .com. Hook up with our social media to see the progress on this. If you're interested in what we're doing, I think that, one of the things about American Gene Technologies that's unique is that I'm taking everything I learned from the software industry and applying it to biotech. It doesn't seem like anybody else has done that. And what is the most efficient way to develop software? It's to have a tool set of reusable components that you can mix and match to create a myriad of solutions. Ultimately, I believe that gene and cell therapy, and this is what we're, progressing towards, because we have a tremendous amount of foundational technologies now that are reusable in hundreds of different diseases each, right?
Lego blocks, so to say.
It's like the iPhone of, of disease. That curing a disease may be like writing an app one day. This will also bring it all over the world where it'll be garage industry. You guys could cure anything too by using our operating system.
I hope we live to see this day.
Well, I think, if you live 20 years, I promise you, you'll see it. It may be obvious even shorter than that.
Okay.
So, but thank you.
Good. Thank you as well. And also big thank you to all the participants, be it the panelists, attendees, and organizers. Big thank you for this wonderful day. It was exhausting and it was even more exciting and inspiring. Really big thanks to everybody. And now we can wrap it up and we meet again at the next edition. Thank you all.
Thank you. Bye-bye.